So today I'm going to talk about one of the most basic biological processes, cell growth, that has been very intensively studied because when it goes awry, cells become cancerous. And accordingly, it has a very, very long history. It's one of the first biological processes to be studied, starting with For some reason, my slide switching doesn't seem to be working. And it was working a minute ago, but I guess we can use that. Starting with the work of Jacques Monod. And one of the things that he did very different from his contemporaries and that made his work particularly noteworthy is that he wasn't satisfied with seeing that the more food you give to the cells, the faster they grow. But he actually quantified this trend very rigorously. And two other contemporaries of Jacques Monod, Crabtree and Warburg, observed that the faster the cells grow, the more they tend to ferment. And this was a very surprising observation. Now it's known probably, as most of you are well familiar with, the so-called Warburg effect. It was very surprising because when cells grow fast, you assume that they need a lot of energy. And why should they use a less efficient method to generate energy when they have plenty of oxygen, but they would ferment? And here is sort of a modern data measurement to show you what this effect is all about. When cells grow very slowly, the amount of oxygen that they take in is about equal to the amount of carbon dioxide that they produce, which means that they grow based on complete oxidation of the glucose in a fully respiratory regime. And then as they grow faster and faster, at some point there is a dramatic bifurcation. A break point after which, as they grow faster, they tend to consume less and less oxygen, and they begin to ferment a lot. Bless them. That's how they make ethanol and beer, wine in the case of yeast, and that is how cancer cells generate a lot of lactate in the case of human cancers. And it is not clear to this day, almost a century after this was first observed, why the hell cells would do that and involve in what appears to be a less efficient metabolism when they grow very fast. And you can frame this question in a much more broad conceptual way. If you have two parallel metabolic pathways, how do you decide which one to use? What are the trade-offs? And what do you gain from one or from the other? And what I'm going to do is I'll use new technology to address these very old questions. Some of them are more than a century old. And I'm going to keep the quantitative tradition of the pioneers who first studied them. And just one more sentence in the way of introduction in terms of what was my philosophical motivation to undertake these studies. Because cell growth is such a basic process that has been studied for such a long time, one may expect that we know a lot. And it is true that we know a lot of the details of the basic metabolic pathways, but how these interact together dynamically in time and in different environments, we know actually very little about. So I think this is a really good case study where we can take what we already know at the level of details and sort of shoot for the more systems level understanding. And conveniently enough, cell growth is among the most conserved biological processes in all of them. So what we learn from the cell growth of simpler eukaryotes, we can perhaps extrapolate to more complex eukaryotes. And there are two main questions that my experiments addressed. One is how cell growth adapts to a dynamic environment, and this is what I'm going to talk mostly about today. The other one, I don't have much time to present. Depending how fast they go, I may hint a little bit about it. And this is the trade-offs of this switch of aerobic glycolysis that I mentioned early on. Again, aerobic glycolysis, just to reiterate, to remind you, the terminology can be confusing, is when cells ferment glucose to either lactate or ethanol, even though they have abundant oxygen. Okay, so here is the experimental design in the system that I'm using. This is essentially a very complex test tube that allows me to control very, very precisely all the growth parameters, do very, very well-controlled measurements. I can take samples without disturbing the cells. What it is a bioreactor in which I can grow, in my case, budding yeast. in well-aerated, well-stored conditions. And one measurement that I want to take a minute to explain, because it's not very common, is measuring those fluxes of oxygen and carbon dioxide that I mentioned at the beginning. What I have is, I know I aerate the culture at a constant rate. I know exactly what it is with 0.1% precision. It's exceedingly high precision. I know what is the concentration of oxygen and carbon dioxide that goes in. I can measure what is the concentration that goes out. And then it's a question of very simple mass balance analysis to compute what is the flux of both oxygen and carbon dioxide. Very simple. And here is the first measurement that I'm going to show you, because it's the most conventional, and probably most of you have done something of this sort. Here I'm just measuring biomass density, and as a measure for biomass density I use optical density. Optical density at 600 nanometers is essentially how cloudy the culture is. And you can see very well the two phases of growth. First, cells grow very rapidly, and the party is a lot of fun. The music is loud, and then the music stops suddenly and completely. And for 5 hours, they don't grow at all. There is a complete cessation of growth. And after that, they pick it up slowly, and they start growing again on the ethanol that they produced during the first phase. Now, this is all very old. In principle, the qualitative result here is all very old. This is what the contemporaries of Jack knew. But incidentally, we still don't understand why the hell they would hit on the brake so suddenly and then they would not grow for 5 hours. It's a mystery that has endured many decades, and I think I'll be able to illuminate that. And then it's pretty interesting here. If you look at this exponential, I'm going to show much more quantitative treatment of that. The cells don't really seem to be slowing down and preparing for anything. They seem to party as much as they can, like a graduate student, and then when they can no longer do it, they prepare for the hardship of what's about to come. OK, so for this exact same culture, I just
have a quick question about this. For the dioxic shift, if you continue to support the cells with additional glucose or glycerol nutrients, will they still have that phase 1, phase 2 growth, or will they continue along with phase 1?
So in principle, in a chemostat, right, so what you could do is you could keep diluting the cells so that you keep the cell density the same, and you keep adding fresh medium, and then you get to a steady state. And then you could actually get to a very stable steady state of aerobic glycolysis when the cells keep fermenting a lot, even though they have lots of oxygen.
OK, but if you keep the density high, like at where the shift occurs,
yeah.
So right there, if you don't change, if you don't dilute the cells, but you somehow keep spiking in additional nutrients.
In principle, yeah, that's right. You should be able to continue doing it. I don't think that somebody has done it in a good quantitative way, but that's certainly the expectation. You should be able to continue it. And I also promised that I'm going to do a lot of other very accurate quantitative measurements, and here is one of them. I'm showing the oxygen and the carbon dioxide. So as I mentioned at the beginning, I'm measuring what is the concentration of oxygen in the exhaust gas. So initially, the biomass density is very low, so we can barely see it. But as the culture grows more and more, they start consuming more oxygen, as you can see it here. And equivalently, as I told you, they ferment a lot. They make a lot of ethanol. As they make ethanol, they produce even more carbon dioxide. And then when the music stops, there is a complete cessation of respiration, almost. And then growth resumes again now on ethanol. On ethanol, it's obligatory respiratory growth. They cannot ferment it. They can only respire. And you can appropriately see that the concentration that they consume more oxygen and produce less carbon dioxide. So if you look at this data in a very qualitative way, that is recapitulating what people knew decades ago. Now, what I'm going to tell you, one of the big messages of my talk, is that if you go and quantify this more accurately, you actually find things that are so surprising that they spend months and months not believing the data and having to measure it in many different ways, and ultimately it turned out to be true. But most of the insights that I'm going to give you actually come from quantifying these fluxes very accurately. And just to continue my description, here at the end, the cells stop respiring because they go into quiescence. So essentially, you see the full span of all growth phases that this simple eukaryote can have. And now if we superimpose things a little bit, the growth, which I showed you before, but now I'm plotting it on a semi-log plot so that you can see the exponential as being a straight line. you can see that in the first phase, growth rate changes very little, as shown by this line in a semilog plot, by the data in a semilog plot lying very nicely on a line. And then you can see the 5-hour period when the cells are agonizing and preparing to grow on ethanol. Here, what they've done is just plotting again the biomass density, oxygen, carbon dioxide, and I'm comparing. You can see qualitatively that the flux of CO2 is much higher during the first phase, and the reverse is true for the second. And here came the big surprise. When I looked at the RQ ratio sounds fearsome. It's really, really simple. It's just the molar ratio of carbon dioxide to oxygen. It tells you how much fermentation they're doing relative to respiration. If it's 1, it means it's complete respiration. If it's larger than 1, it means they ferment a lot. And I was amazed when I saw that this is changing here, because for all my research and all the people whom I know have been assuming that if you have an exponential biomass increase, that requires that growth rate is constant. And if growth rate is constant, I was thinking maybe you're at a steady state. But now if I look at this ratio here that tells me what are the various metabolic fluxes, I see that while the growth rate is constant, the metabolic fluxes are changing, and I'm just going to blow it up a little bit for you so that you can see it. So here again I have this constant growth. Cells are growing as fast as they can possibly sustain. It's full speed on. And this measure of how much they respire and how much they ferment keeps changing in a very monotonic, smooth way. And here, just a quick note to say that when they finally hit the fully respiratory regime, they can only respire on ethanol. What they find for the RQ measure is remarkably close to the theoretical expectations. So that's good in a way validation of my data. But when I saw that, initially I was incredulous. I kept repeating it, and it was very reproducible. And then my skepticism took the better part of me, and I said, I cannot believe something as radical and unexpected based just on this measurement. Maybe there is something wrong with my detectors. And I decided, in this case, I was fortunate because I can do a very similar measurement of those fluxes, respiration and fermentation, by completely orthogonal experimental method. I can just measure glucose and ethanol, and then if I do those very simple mass balance calculations, I can again see, are those fluxes changing? And here I'm showing you the data just from the first phase in the aerobic glycolysis. When the music is loud, the cells are parting, growing as fast as they can. And you can see that the glucose consumed per cell actually goes down steadily. Somehow the cell seems to be more efficient and seem to be able to grow by consuming less glucose. And they seem to produce more ethanol per molecule of glucose consumed, which is again consistent that as toward the end of their growth, they actually boost their fermentation rather than their respiration, which to me is very, very surprising. Why? Because when the cells are growing, they consume their glucose. The glucose concentration gets lower and lower, and you expect them to get into some sort of economizing mode. When they try to spare the glucose, they try to be as efficient as they can possibly be, and you'd also expect that at low glucose they would induce respiration, they would suppress their fermentation. What you see, it's actually the opposite. And I kept doing those measurements over and over again, and that's what it is. So before I go further to try to explain what this observation means, I'm going to show you the same I'm going to show you the result of another technology testing this idea whether when growth rate is constant, metabolism could be changing and the whole growth strategy. Because here I showed you using two different, based on two different experimental approaches, but I'm actually still skeptical. I find this just unbelievable. And I used gene expression microarrays to measure it. And here I'm just giving tribute to the first use of this technology by Pat Brown and collaborators. And in fact, this technology was inaugurated on this very same problem. They measured growth of yeast in glucose medium. And on those first microarrays. This is the type of analysis that they were able to do to validate the technology. But now I want to have a much higher time resolution and much more using the much more mature and accurate technology to be able to see, are there gene expression changes associated with the changes in metabolism that I have been observing. And this is what they see for the first phase of very fast growth in glucose, aerobic glycolysis. So again, this is a semi-log plot, and the straight line indicates perfect exponential growth and constant growth rate at 0.4 per hour, the fastest that the cell can sustain. And you can see that about 1,500 genes have monotonic trends, either go down or go up, while the growth rate remains constant. And this really reiterates and drives home the idea that seeing constant growth rate and the culture growing exponentially does not indicate that the culture is at steady state. A lot of things could be changing under the hood and still keeping the highest growth rate that the cells can sustain. And another example of this is, remember when they recover, when they get hammered and they don't grow for 5 hours, then they recover and they start growing more slowly. And here I'm capturing this period when they grow about an order of magnitude more slowly, again exponentially. And you see the same thing. Again, another set of, in this case, 2,000 genes are actually changing monotonically while growth rate remains constant. And I'm just going to go for some of the low-hanging fruit because I don't have time to. Yeah, go ahead.
Are those the same 2,000 genes?
There is a substantial overlap, more than you would expect by chance alone, but they're not the same by and large. They're about what is the a third of them are common between the two. Now, I don't have much time to go in details, but I'm going to go for the low-hanging and the simple fruit because I'm talking here about cell growth. So cell growth, to first approximation, two of the very basic processes would be making biomass. You got to synthesize protein, and I'm going to look at ribosomal proteins because that's the basic machinery that's making all the rest of the protein. And then, of course, cells have to divide. And then I'm going to look at the cyclins for the cyclin-dependent kinases. Those are the engines of the cell division cycle, and they're particularly nice targets to look for in transcriptional data because it is well known that cyclins are transcriptionally regulated. That's part of their main regulatory mode. And here I'm showing the data for the large ribosomal proteins that they have just color-coded to show that there are two distinct classes. And some of them are expressed very highly during aerobic glycolysis, the fast growth, and they go very low when cells get hammered and go up a little bit, so on. Those are much lower to start with during aerobic glycolysis, the red guys, but then they're expressed at higher level than the green ones during the growth in ethanol. And to me, that was very surprising. I didn't know how to think about it until I saw that many of the members here, if I see a green gene, its paralogous protein that arose from the genome duplication is going to be in the red side. So let me show you a typical example. So we are looking at two proteins that have extremely similar sequences. They all arose from the genome duplication. And one of them is expressed highly during aerobic glycolysis, goes down. It's not expressed very highly during growth in ethanol, and the reverse is true for the other paralog. And if you think about, again, this part here first corresponds to fast growth, and this is the slow growth in ethanol. This is quiescence. It makes sense when cells are quiescent, they're not expressing ribosomal proteins. So if you think about what this means, if this is translated at the level of proteins, it means that you're actually changing the distribution of proteins in the ribosome and the constitution of ribosomes, and this is something that we are actively testing using mass spec currently. But one way to think about it is that there are 79 or so ribosomal proteins that can participate in building up the large ribosomal subunit. And the large ribosomal subunit contains only 43 or so. So the paralogs may substitute for each other, and depending on which paralog you have, you might have different translational specificity, or so is my hypothesis, which is not confirmed yet. It's just an intriguing one. And here I'm showing that this pair is not unique by any means. There are a lot more pairs that do essentially the same thing. And the other low-hanging fruit that I promised I'm going to look at are the cyclins for cell division. So here I'm showing two pyrologous ones, the PCL1 and 2. Those are the major cyclins for the 485, the cyclin-dependent kinase that regulates a lot of metabolism. And you can also see, using this probably more obvious plot, the lines. The lighting is not very good, so it's hard to see the colors. But you can see that PCL2 is higher during growth on glucose, and PCL1 is higher during growth on ethanol. And again, this suggests that different cyclins are being used. And the same is true for cyclin 1, 2, and 3, which are the cyclins for CDC28, which is the main cyclin, the main cell division cycle, cyclin-dependent kinase. Okay, so I promised at least to give you some speculation as to why the cells are able to reduce their consumption of glucose toward the end of their growth while still keeping their growth rate constant. And this provides a very strong clue to that. Now, if you look at the metabolic data, if you normalize the oxygen consumption per unit biomass, which is very easy, I've measured both, you see that the oxygen consumption actually goes down. And one way to think about this is that if the oxygen consumption goes down, perhaps the cells are making much less reactive oxygen species, so they need to detoxify fewer reactive oxygen species. And this data here for the two genes that catalyze reactions producing most of the NADPH is consistent with this hypothesis. Now, why is NADPH special? I should take a minute here to explain. Probably you are familiar from your biochemistry class with NAD and NADP. And NAD is the major cofactor that carries around electrons for everything that has to do with energy production in the TCA cycle and so on. NADPH is very special because there are very, very few reactions that can produce it in the cell. And it is the only source of reducing equivalence which you can use for either biomass synthesis or for detoxification of reactive oxygen species. So that makes it convenient to monitor what the hell is happening with the NADH. And in this case, the cells seem to be producing substantially less of those enzymes, which is consistent with the idea that what they're really doing is reducing their respiration so that they can alleviate their burden of having to deal with detoxification of ROS. And how am I doing on time? How much?
5 more minutes.
Right. OK, that's great. So I'm going to have the remaining 5 minutes for questions. The second part of my talk, which I don't have time to give you, essentially shows data that I think conclusively supports this idea that when cells do more respiration, they actually, I'm going to summarize it for you in words. I don't have time to show you the data. But when in all cases that I have profiled experimentally, when cells consume less oxygen, they seem to need much less energy compared to cells growing at the same growth rate who ferment more, which suggests that oxidative phosphorylation can be very lucrative. You make a lot of energy, but also has a really high overhead. It's very pricey. You got to pay the price for it because even though we got all this great ATP, now you're going to use some of it to deal with all the damaged protein and recycling. And there is recent work to suggest that the recycling is actually very expensive. But what I showed you today, just to recap, is that the growth rate can remain constant while the major metabolic fluxes and gene expression change. So if you see exponential growth or constant growth rate, this really does not have to imply steady state, not in a strong way. And aerobic glycolysis can reduce the energy requirements for cell growth, which is supported by this observation that toward the end of the first growth phase, the cells were keeping their growth rate by consuming less glucose per unit cell, per unit biomass. And today I spoke mostly about metabolic and growth dynamics during many life cycles on a very long time scale. But most of my work actually focuses on dynamics during the life cycle of single cells. And in this case, we also find that steady state is, in fact, a very poor approximation. If you go to the level of single cells, even single cells taken from steady state populations growing in chemostats, you find that most metabolic processes are very, very dynamic. Of course, you know that because of the cell division cycle, you're going to see some dynamics. But those dynamics are not limited just to cell division, DNA replication, and so on. They extend to pretty much all of metabolism and biomass synthesis. So with that, I'm going to thank the people who made this work possible. On the first place, Alex, my supervisor at MIT, my mentor. Of course, David Botstein, who is in many ways ideologically the person who showed me how little we know about cell growth and how much potential there is to expand our knowledge based on new technologies there, and my collaborators at Harvard. So I hope you have lots of questions. Yeah?
So you showed the different graphs of what was happening for gene expression profile changes during the different growth phases.
Right.
Did you have one of what is happening during the interim quiet period?
Oh, yeah, I have that. I did not include it just for brevity. It is really interesting. So I'm very glad you asked for this. So if you look here, there is absolutely no biomass increase for 5 hours. And you may assume that the cells are quiescent. And in fact, I was curious to see that. You see that, in fact, gene expression here is very different from gene expression here when the cells are truly quiescent. A lot of the changes go in the same direction. For example, ribosomal proteins go down in both cases, here and here. But here they go down by a lot more. So, and here there is some specific gene expression which seems to be preparing the cells so that they can resume growing on ethanol. So if I'm to make my speculation to explain why the cells are having this major crash and need a lot of time to recover, is that here, while in the gene expression data, in some of the metabolic data, you can see signs that the cells are doing a little bit to prepare for what's coming next. They actually do relatively little of that. It seems that what they're optimizing is using the last bit of music to keep the party loud, to keep growing as fast as they can, and they don't do enough preparation for the next fully respiratory growth. So a lot of that happens here, and in fact, if you let them grow for a very long period of time so that they get reassured that party is going to last forever, then they get really hammered and this gets longer. You can actually regulate that. But to summarize my answer to your question, you see a lot of changes that are associated with no growth. Some of them are related to cells being similar to the ones that you see in quiescent cells, but smaller in magnitude. And you see quite a lot of gene expression changes that can be interpreted as preparing the next phase of growth. Yeah?
How sensitive are these trends or the gene expression observations that you see to the media that the cells are in and the relative ratios of substrates or oxygen?
So oxygen.
The same if you are in completely different.
That's right. That's right. I've done this experiment in many different media, different biomass densities, and qualitatively the trends that I see are identical. Quantitatively, you can see some differences, but what I was going to say something really interesting, but that's right. They're not very sensitive to the media. You see the same on different media as well.
So if you provide different substrates other than glucose, do you see the kinetics of the initial growth change?
That's right. So glucose is their favorite. So any other substrate is less preferred than that. So depending on if it's a fermentable sugar, you're going to see something similar in terms of the diuxic shift. If it's not fermentable, of course, you're not going to quite see that. In this case, I started with glucose because I really wanted to understand aerobic glycolysis, and you see aerobic glycolysis being full-blown when cells grow very fast on glucose. And just to go back also to your question, because I measure oxygen in my environment, in the exhaust gas, I can really know that the cells are not limited on oxygen, and this is just a very, very clean way to show that in conditions of abundant oxygen supply, cells would still choose to ferment, which is always a bone of contention when it comes to a study of human cell lines, and especially cancers, because it is much harder to have the same level of quantitative accuracy. In this case, my error bar here, I have about 1 million data points on this plot. My error bar is 0.001%. And I know they've got plenty of oxygen, but nonetheless, they opt for fermentation.
You mentioned the connection to cancer. I was just wondering, you know, we think of cancer as being, I guess, correlated to the second phase that you see, right? But
this one here, the first one, that's right, that's right. Yeah, the first one is essentially very fast growth. It is full gas on.
Oh, I see. So does cancer ever have a phase that's corresponding to the second phase that you see?
With cancer cells, essentially yes. There's very close correspondence which you can find to all the phases here. The experiment is hard to do in the same way and as cleanly as you can do it with budding yeast, and that's why we use a model system. But otherwise, pretty much everything that you measure in yeast, you can find correlates of that in cancer cells.
So I mean, like, is it like, oh, a later, like, you know, like does this have implications for whether the cancer cell is, you know, in human and how we study, how we think about its metabolism versus like in a culture dish where just, you know, it's growing so rapidly, not limited by like.
Oh, absolutely. I think that very many biological findings have to be understood only in the context of the proper physiological conditions and cell growth. Now, if you're studying something that is very universal and genetic, perhaps the growth conditions are not as essential. But nowadays, as studies become much more sophisticated and we have better and better technologies, I think that more and more of our results become highly contingent on the growth conditions. And I think it's a very good idea to take those into account and to especially when comparing results between different experimental studies.
So I think if I understand it correctly, what your data shows is that as sort of the concentration of glucose decreases, cells actually use even more fermentation.
That's right. That's right.
Can you explain it again?
That is very surprising to me. I did not expect it to say it. I can show you with 3 different experimental methods that this is what happens. What my hypothesis is and what my interpretation is, and most of that is actually based on what I don't have time to present you, but my interpretation is the cells have evolved, at least in the case of budding yeast, to try to keep the highest growth rate that they can possibly sustain given the changing conditions. And here they have been growing for many generations in high glucose media, and they have achieved their highest growth rate, and now they don't want to slow it down. But as glucose gets exhausted, they have to decrease their flux of uptake of glucose. They just cannot keep uptaking the same amount. And the change in their metabolism actually enables them to grow more efficiently. So if you look here at glucose consumed per cell, which is normalized to the biomass density, it is going down. So the change of metabolism is actually enabling them to keep their highest growth rate possible with smaller flux of glucose. And at least one component of the cells being able to do that is that they can significantly cut on their expenses to deal with the consequences of respiration, which is going to be more reactive oxygen species and damaged proteins, which is my interpretation. I always like to separate things that I can show without a tinge of doubt and things that I interpret. So what I'm absolutely confident in is essentially what the data show, that the cells can become much more efficient in terms of keeping their growth rate by taking up less glucose. And I can also show that in conditions of steady-state growth, there is a very strong, essentially perfect observation that in every single condition, when cells respire more, they also seem to need more energy to grow at the same growth rate. And if I apply this same principle here, it is intuitive and simple to extend it. I can just say that what is really happening here is that the cells are not boosting their income, they're reducing their spending. They become much more linear, and if you think about it as a metaphor of a race car, you can make a car go faster either by bulking up the engine or by reducing the weight of the car. What the cells seem to be doing here, is reducing the weight of the car rather than bulking up the engine.
Is it fair to say that fermenting cells can make more biomass from less glucose than respiring cells?
In this condition here, which is dynamic or transient condition. That seems to be the case.